Very good morning to all. So my job is very simpler as the previous talk has in detail covered a lot of, all the aspects of the ctDNA analysis. So in this talk, I will be, the plan of my talk will be, I will be just going through few slides showing how there was a paradigm change in the lung cancer diagnosis as well as management, few points on the gene fusion, various platforms, then the pre-analytical factor, which is very important when you deal with ctDNA analysis. And then finally, the few studies in literature who have documented fusion gene in ctDNA in lung cancer. So we all know how there is an evolution of lung cancer care. In early 2000, the broad classification of lung cancer as small cell versus non-small cell would suffice. Then came an era where you need to histologically accurately classify adeno versus squamous. And now is the molecular era where you need to pinpoint which exact molecular targets are present in a particular case. So for each histology-based subtypes, there are different spectrum of molecular targets. And now there is a ever increasing list of FDA-approved drugs. So this makes it a point that the molecular characterization has to be performed using highly sensitive, specific, adequate, and validated tests. No matter what tumor samples, histology, cytology, cfDNA, low percentage of tumor cell, what is the answer? You have to provide a molecular characterization of a tumor. So what is the, so how do we go about it? So multi-gene testing, that is the NGS panel, which should encompass must test biomarkers, should test biomarkers and other investigational biomarkers. This should be your panel when you deal with any multi-gene testing. So coming to fusion genes. So what are fusion genes? Fusion genes are basically juxtaposition of two independent genes which come together, and following which there is this driver mutation, this driver gene, molecular driver, which stimulates the downstream signaling pathways. So for a fusion gene, it can be either a translocation, deletion, inversion. It can be within a same chromosome or within a different chromosome. So when it comes to lung cancer, we have many success stories of fusion genes, that is ALK, ROS, RET, NTRK. With ALK, which is seen in about 2 to 7% of NSCLC cases. And now we have like a progression-free and overall survival as close as 5 years with this ALK TKIs. Same holds true for ROS, which is seen in about 1 to 2% of lung cancer cases. And you have around 25 to 30 types of gene fusion partners for ROS. So the ROS TKIs like crizotinib and entrectinib, the latter with a good CNS permeability. So it is very important to identify all these fusions. RET fusion with the commonest partner being a kinesin family. And you now have this FDA approval for the selpercatinib and prazeltinib approved for RET fusion genes. Same hold true for NTRK. You have NTRK 1, 2, 3 fusion genes, and you have different TKIs for this. So with this development and approval of new drug targeting all the rare fusion, it is very important that you do deep molecular characterization of lung cancer specimens. So when it comes to platform of this detection, you have single gene based testing and multiplex testing. With fusion for ALK and ROS, single gene based testing is fine. Like you have very well established IHC markers and FISH. But for RET, NTRK, the only answer is multiplex gene testing and next generation sequencing. So I will spend some time on this slide. So NGS for fusion analysis. When you look at this, you will divide it into various types. So first is how do you analyze, level of analysis, whether it is RNA based or DNA based. Our DNA we all know is very stable. So when you use DNA based analysis for fusion testing, apart from fusion, you also get other details like you get SNVs, copy number variations, MSI, all other details is possible. The only thing when you use DNA based thing is if you have multiple breakpoints at the intronic region and you have large deletions, it won't be able to pick up by this DNA based. As against RNA, RNA is very sensitive for any fusion genes. You will be able to identify and quantify the fusion transcripts irrespective of where is the breakpoint in the intronic region. However, RNA is very unstable. very impure, especially in diagnostic settings when you are dealing with FFP samples. So you have to be very particular about your quality checks before you start with any NGS for fusion analysis. Another thing is, what is your panel target, whether it is a limited gene assay or whether it is whole transcriptomic or genomic. Secondly, when you look at the chemistry of the sequencing or with the library preparation, so whether it is hybrid capture or amplicon sequencing. So hybrid capture, with the help of specific probes, you will target the entire gene of interest. So that is very good when you want to design large gene panels. It also helps you to identify known as well as the unknown fusion partners. So it is very good. It can be used in RNA as well as DNA. But when we are looking at the amplicon based sequencing, so in amplicon based, you will amplify the region of interest. It is mainly pertinent to RNA based testing, and you should have the known and known partners when you deal with amplicon based sequencing. Another is sequencing chemistry, whether you are using ion torrent or sequencing by synthesis chemistry, that is Illumina. Another thing is then depending upon the bioinformatic pipeline. So all these parameters are very important before you test for any fusion gene analysis. We should be aware of this chemistry and these technicalities. So all this is very well established in the solid tissue. However, as we all know, like majority of the cases, like 60 to 70% of lung cancers are in the advanced and metastatic stage, and the only available tissue with you is a biopsy, which is also barely representative of the intratumoral heterogeneity. Also, up to 10 to 20% of biopsies are inadequate. So what is the solution for this case is the non-invasive alternative, that is the cfDNA. So when it comes to cfDNA, what are the recommendations? Whether the patient is treatment naive or on progression. So for first and foremost important, for newly diagnosed patients with advanced NSCLC, plasma ctDNA is a valid tool wherein results are often complementary to tissue analysis. Directly, you cannot start with cfDNA. However, at baseline, if the tissue is inadequate or inaccessible, you can start with this. At the time of acquired resistance, however, the guidelines say that the plasma first approach is preferred, and only if negative, you can do a repeat tissue biopsy. And testing for plasma ctDNA should be performed using a clinically validated NGS platform, and not to use single gene based PCR approach when we are dealing with this. So with this, I will highlight few important points on circulating tumor DNA. So what is circulating tumor DNA? It depends upon the necrosis and apoptosis of the tumor, and then the tumor cells are shed in the circulation. The fraction is very low. So it is less than 0.1%. And also the DNA fragments that are released are very small and they have a half short life. Now, why this is important is that, so like finding a needle in the haystack. So few points on the pre-analytical parameters. Like when you take the sample, whether you are using a EDTA tube or using your Streck tubes. So when you are using a Streck tube, up to 3 days, it's fine at room temperature. But when you are using EDTA tubes, you have to extract the plasma within 2 hours. Otherwise, what happens is that there will be degradation and there will be further dilution of your tumor DNA with the wild type DNA. Another thing is how do you process? You should not freeze the blood sample. Then the spinning should be very fast double spin when you want to extract. So why this is important? When you want to extract cfDNA, this double spin high speed centrifugation is must. Whereas if you want to extract RNA, you should not use Streck tubes, and also there should be no high speed centrifugation. It should be a platelet rich plasma. So all these points are very important from technicality before you interpret any case as positive or negative. Other thing, of course, the depth of sequencing and what technique are you using. So when it comes to ctDNA, what is the main issue is the low sensitivity, but definitely a high specificity of the. So what is like low variant allelic fraction? Even if 0.2% allelic frequency, will consider it as positive. Another thing is contamination with the wild type DNA. You have this chip issue, that is the clonal hematopoiesis of indeterminate potential. All these things, there is very inconsistent shedding of tumor DNA. Majority of patients will have very low tumor DNA. So comparatively, everything you have to take into account before you deal with analysis of cfDNA. So coming to the gene fusions on plasma samples. So in literature, so in our lab, we are not doing gene fusions on the plasma samples. So whatever I will be showing is the data which is published in the literature. So different NGS panels have been used. So mostly they use this hybrid capture panel. So whatever is the majority of the studies, they make use of this Garden 360 and Foundation 1 liquid. So both these panels are hybrid capture. They have around 70 gene panels, of which has SNVs, CN copy number variations, and fusion of approximately 6 genes. Also, they have some indels analysis in some of these genes. So this is one of the largest prospective study which was done on the cfDNA Nile study, that is the non-invasive versus invasive lung evaluation. So in this, they had taken biopsy proven previously untreated non-squamous metastatic NSCLC over a duration of 2 years. And what did they demonstrate is that, so there were some cases which they did upfront tissue and also cfDNA. So overall 95% patients were fully assessed for all 8 guideline recommended biomarkers in cfDNA. So 268 cfDNA sample versus 51 tissue. So that is a very huge number. So 48 cases were positive for the mutations in both tissue as well as ctDNA. The positive predictive value of the genotyping in cfDNA was 100% with a greater than 98.2% concordance. And now the important thing is, when the primary modality was tissue based, only 67% of the patients were identified by tissue as against 87% when cfDNA was used as a primary modality. So this study demonstrated the non-inferiority of cfDNA based versus standard of care tumor tissue based genotyping. So this was a very important study, Nile study. And most importantly, the TAT. It was around 9 versus 15 days. And when it comes to NGS, TAT is a very important key quality metric. The second study was the clinical utility, where they just looked at the ALK gene rearrangements in the cfDNA. And they found that it was a study of around 90 cases of ALK gene fusion, and they were able to identify fusion partners. Then even some cases, the tissue was insufficient, but cfDNA had good results. And follow-up of 3 of these 5 tissue negatives showed response to ALK TKI, which is again an important. With cfDNA, they were able to identify this ALK resistant mutations because it had all DNA-based variants also. And in 6 patients with known EGFR mutation, ALK fusion was identified on progression in 5 of these cases. Similarly, there was a Chinese study. Again, they used a different panel targeting 168 genes using 50 nanogram of ctDNA. And it was blood samples from 24 ALK gene rearranged, and they were able to identify in 19 out of 24 patients with a baseline specificity of 100%. So with all these studies, we see that though the sensitivity is lower, but specificity is 100%. And so there were also few studies which didn't use hybrid capture, but also amplicon based sequencing on ion torrent platform. So again, the same with a very low nanogram, 10 nanogram of ctDNA, still a specificity of 100%. The other thing when it comes to ctDNA is depth is very high. It is around, so if in diagnostics samples in tissue, if you use a 500x coverage with ctDNA, it is 5,000, 15,000, and what some studies have published like around 60,000 depth coverage. So that is what is very important because of very low allelic fraction of the target in this. So to conclude, gene fusion analysis is crucial in the context of lung cancer precision medicine. NGS presents several advantages, and it allows the analysis of more targets, saving time and material if the turnaround time is acceptable. So clinical relevance of ctDNA is now well recognized in all stages of clinical evaluation. But in spite of this interesting available results, the real utility in clinical practice when it comes to fusion has to be assessed on the large scale studies. And what are the 2 main challenges is to have a standardized assays for fusion and to translate these observations into clinically useful test. This I would like to end my talk. Thank you. And I would like to thank all the organizers.
Thank you so much, Trupti, for highlighting the features and challenges in liquid biopsy. This talk is open for the questions. Any questions from the audience? Okay, Thank you,
Thank
Trupti. Yeah.
No, you mean for fusion? No, we are not using fusion. We are using amplicon based in our RNA base. That is why we are not able to assess the fusion gene. So that we are yet to validate. We have not yet. So the panel that we have is not acceptable for this analysis. So that is the thing. So what we are using here, we have hybrid capture for DNA based variants, but the RNA that we are using is amplicon based sequence. That is why with this cfDNA, we are not able to assess.
Okay, one more question, Trupti. You mentioned about the Streck tubes that they don't pick up the RNA fusions. So what about the samples coming from outside? Because for the transportation, we need to use the Streck tubes. So
No, I mean cfDNA can be extracted, RNA.
yeah, if it is a RNA based, your test is there.
EDTA is fine.
Yeah.
Tube is specifically not
But
designed
again, for the transport, it takes
Oh, yeah,
around
that is
48 hours. So do you reject those samples if they come to you in the Streck tube, if it is the RNA fusion based test?
So, ma'am, as I said, we are not
Doing.
RNA
Okay.
based tests. So right now, whatever we are getting is from outdoor stations. We are getting Streck tubes, and we are doing cfDNA. So that is fine.
Okay.
If there is no access to Streck tube, can the any lab or the person can separate the plasma and send it to you? Is it possible? Is it allowed?
Yeah, yeah. So the
That is
thing
acceptable.
within 2 hours, you have to extract the plasma.
Separation and then
Yes,
it
separate
and then
the
what is the
plasma.
stability in the plasma?
You mean that you have to keep in the fridge. Before plasma extraction, you cannot freeze the sample.
Mhm.
After that, you can.
But what is the shelf life? 3 days, 5 days stability?
That I may not be able to
Okay.
tell you after extraction how much it is.
Okay.
Because it will take.
Yeah, because by the time it reaches